In the embryo, a mesonephros develops from the middle part of intermediate mesoderm posterior of to each protonephros. Soon after its degeneration. Posterior to protonephros soon after its degeneration. At first, new mesonephric tubules join the existing pronephric duct and are segmentally disposed. Later on, the tubules multiply by budding so that their segment arrangement is disturbed due to increased number of should be distributed due to increased number of tubules per segment. Tubules of pronephros or mesonephros develop similarly and homologous. However, mesonephros is functionally better than pronephros because mesonephros are more numerous, longer, and develop internal glomeruli enclosing capsules forming Malpighian. Thus, they remove liquid waste directly from glomerular blood rather than indirectly from coelomic fluid, as in pronephros. Pronephros is also termed Wolffian body. With the disappearance of pronephros, the old pronephric duct becomes Wolffian or mesonephric duct. In amniotes, reptiles, birds, and mammals, mesonephros is functional only in the embryos, replaced by metanephros in the adults. Fishes and amphibians, mesonephros is functional both in embryos as well as adults. Okay, so in sharks and sea lions, tubules extend posteriorly throughout the length of coelom. Such kidney is sometimes called posterior kidney or opisthenephros. Whereas in adult annuitants, udales, and embryonic amniotes, the mesonephros does not extend posteriorly. Mesonephric kidney in mixed units, we don't need that. Metanephros are highly specialized, formed in the posterior portion of mesoderm. Mesonephros degenerate except in gonads. Metanephros are highly convoluted, larger, not wanting nephron, and ureter from Wolffian duct, urinary bladder store urine. So here ureter is formed from Wolffian duct. Okay, so here you can see the structures. Let's go further. Testes and male genital ducts. Testes of vertebrates are paired organs of moderate size, usually found attached to kidneys. Testis is compact gland covered by coelomic epithelium and seminiferous tubules, composed of numerous highly coiled embedded in connective tissue. Tubules are lined with germinal epithelium, which gives rise to billions of sperm. On maturity, the sperm are set to free in the lumen of tubules and move towards the genital ducts. Okay, sperms are released in the coelom from where they pass through abdominal pore located at the posterior part of coelom, into which the two are elongated bodies. In most amniotes, the opisthenephros is differentiated into anterior genital and posterior renal portions. In the anterior genital portion, in males, some uniferous tubules lose excretory function form slender vasa efferentia and become continuous seminiferous tubules of the adjacent testis. They serve to convey sperm of testis to the mesonephric duct of kidney. Thus, in male amniotes, mesonephric Wolffian duct forms a urinogenital duct, serving both as a vas deferens for sperm as well as ureter for urine. However, in many elasmobranchs, external ducts drain urine from kidney to cloaca, so the mesonephric serves entirely or mainly as a vas deferens. In embryos of anura, each testis is made up of two portions. Male frog, the anterior portion disappears and the posterior portion becomes the adult functional testis. In adult male toad, the anterior portion also persists. The Bieder's organ contains large cells similar to mature ova. In male amniotes, a metanephros develops as the adult functional kidney with its own urinary duct or ureter to transport urine. You can see in male amniotes there is vestigial Mullerian duct, and you can see the vas deferens. Also the ureter. So the adult functional kidney with its own urinary duct or ureter to transport urine. The mesonephric or Wolffian duct becomes solely a genital duct or vas deferens. The remnants of embryonic mesonephros and a coiled portion of mesonephric ducts become epididymis of the adult kidney, from which testis sperms pass through epididymis, then through vas deferens to reach urethra. So there is epidermis, testis from epidermis, vas efferentia, testis. So here from each testis sperms pass through epidermis, then through vas dehiscence to reach urethra. Vas deferens, and then here is the ureter, urethra. In most mammals, testes descend permanently into extra-abdominal skin bags called scrotal sacs. In rabies rodents, they are lowered into sacs and retracted at will. Passage between abdominal cavity and scrotal sac through which descends is called inguinal canal. However, some mammals such as monotremes, insectivores, elephants, whales, etc. legs or scrotal sacs so that the testes remain permanently abdominal like ovaries. Copulatory organs. Copulatory organs are absent in amniotes since they have usually external fertilization, but in amniotes fertilization is internal and preceded by copulation or mating. Male amniotes develop copulatory organs for transferring sperm into the genital tract or females during copulation. They are particularly characteristic of reptiles and mammals. In Asperbanks, bases of pelvic fins are modified as claspers. These are grooved cylindrical structures that are inserted into the female cloaca to inject sperm. In dogfish and some allied forms, there is blind muscular sac called siphon located at the base of clasper. This sac gets filled with water, which is used to force the spermatic fluid into the cloaca of female. In Sabertelius, the anal fin is modified as a gonopodium for sperm transport. It is modification of anal fin. Snakes and lizards have a pair of retractile, grooved and sac-like hemipenis which can be inverted through cloaca. This retraction is controlled by modification of body of musculature. Classical and some birds and prototherian mammals have an unpaired grooved and erectile penis formed as a thickening of cloacal floor. The higher mammals have a true external erectile penis with a tubular groove contains with a spongy urethra. Series of excessive sex glands associated with penis secrete a fluid in which sperms are carried. Ovaries and female genital ducts. Female amniotes ovaries are large, occupying much of the body cavity, produce thousands of eggs as fertilization external. In amniotes ovaries produce fewer eggs because fertilization is internal. Ovaries of reptiles and birds is still large and the eggs produced contain much yolk. However, mammalian eggs contain very little yolk so that the ovaries also remain quite small. Ovaries are generally pair structure, but only a single maiden ovary occurs in cyclostome. Okay. So you can see here the structures. In male amniotes, testes connected to the kidney. There's Ilfian duct, which is transporting both the sperm and the urine. In male amniotes, the Ilfian duct and the ureter is different. Similarly, female amniotes, it is called the Mullerian duct and the ureter is different. Female amniotes, there is the Mullerian duct and also the Ilfian duct, both present, and they are separated. So Mullerian duct is still present in females but vestigial in males. Then we will do the Mullerian duct short note. So this is done for now.